Yes. So in anaerobic ammonia oxidation or anaerobic ammonia oxidation, here the ammonia is used by the chemolithotropes as an electron donor, and they reduces the nitrite. So they are reducing the nitrite. So usually, just a minute, whether I have started recording. Just एक second फक्त. Okay. So the reaction carried out is ammonia in presence of nitrite. This is your NO2 nitrite. This ammonia is used as an electron donor. So the two electrons are flowing to the nitrite, and the nitrite is reduced to N2, and two water molecules are given out. So the Gibbs energy is free Gibbs energy is minus 358 kilojoules per mole of one ammonia. So this particular reaction is taking place in the chemolithotropes. So these have not been isolated in the pure cultures because this particular environment cannot be done. Yes, this environment cannot be usually done in a laboratory conditions, and therefore you cannot isolate it in the pure culture form. Because every favorable conditions has to be given in the laboratory, and therefore this condition is not cannot be satisfied in the laboratory, and therefore you cannot isolate it in the pure culture. But they have been enriched in a reactor by using wastewater containing high concentration of ammonia, NH4 2 plus, in a process known as anaerobic ammonia oxidation, that is Annamox process. So based on 16S ribosomal RNA gene sequencing, the microorganisms which is having the ability to undergo such type of reaction, they belong to the genera Brocardia, Anammoxidans, then Pneumania, Studgratiensis, Scalendula, Brode, Scalendula wagneri. So these are the microorganisms which are having the capability of undergoing the nitrite reduction in presence of ammonia. A nitrifying bacteria like Nitrosomonas europaea, it oxidizes the ammonia under anaerobic condition using nitrogen dioxide. They are using nitrogen dioxide as the electron acceptor. So this is the another bacteria which oxidizes the ammonia in anaerobic condition. Now we will see the dissimilatory nitrate reduction. So this is strictly an anaerobic bacteria, because usually the chemoheterotrophic bacteria, they have the ability to reduce nitrate to ammonia, and therefore they are therefore it is called as, see, uh, in dissimilatory nitrate reduction, the nitrate is reduced to uh, that is dinitrogen or N2 gas, as well as in dissimilatory, that is dissimilatory nitrate reduction, it is a strictly anaerobic reaction which takes place in the chemoheterotrophic bacteria, which reduces nitrate to ammonia. So these include Dinitrovibrio acetophyllus, then Thiora selenates, Volinella succinogens, and Disulfovibrio gigas. So these are the microorganisms which bring about the dissimilatory nitrate reduction. Now, since these microorganisms, they generate a proton motive force. See, what do you understand by the proton motive force? Proton gradient is created in the periplasmic space, and because of the proton gradient force only, there is a synthesis of ATP molecule. So usually this process generates a proton motive force and is not inhibited by the higher concentration of ammonia. So the reduction of nitrate to ammonia is regarded as dissimilatory nitrate reduction, or this process is also called as nitrate ammonification, because from nitrate you are getting the ammonia and therefore it is called as nitrate ammonification. Now, the last point is your fermentative nitrate reduction. Now, certain microorganisms like Clostridium perfringens and propionate producing anaerobic microorganisms, they reduce the nitrate to ammonia as in nitrate ammonification, but this reaction is not coupled to the generation of proton motive force. See, in nitrate reduction, that is ammonification, nitrate ammonification process, there is a generation of proton motive force. But in here, there is no generation of proton motive force because it is a fermentative process, and Clostridium perfringens and propionate, that is propionate producing microorganism, which are strictly anaerobic microorganism, reduces the nitrate to ammonia. So since electrons are just disposed of, means whatever the electrons are that is eliminated out, they are just disposed of in nitrate reduction, and the electron acceptor is acetyl CoA, and therefore this acetyl CoA is converted to acetate, which is synthesizing the ATP molecule when it is entering into the Krebs cycle. Okay, so these fermentative propionate producers, they do not produce propionate. Now, in this case, the propionate producing anaerobes, they do not produce the propionate in presence of nitrate, but they produce only the acetate. They produce acetate from the lactate, which we'll see in the figures 8.16 and 8.18. So this process is referred to as fermentative nitrate reduction, and the nitrate in this case is termed as the electron sink. See, because the electrons are dumped off, because these electrons are just dumped off, it is not generating the proton motive force, and no ATP synthesis is taking place by using the proton motive force. These electrons are just dumped off, and therefore it is called as the electron sink. Though it is well known that oxygen represses the expression of denitrification enzyme genes, as you know that this particular anaerobic that is Clostridium perfringens and propionate producing bacteria is strictly anaerobic in nature. So oxygen will surely repress or inhibit the expression of denitrification enzyme. So aerobic nitrate reduction has also been known, but you know that aerobic nitrate reduction is also known. So usually it occurs in certain facultative anaerobic bacteria isolated from anaerobic digestion, and these bacteria belonging to microvigula denitrifications, they can reduce nitrate under air saturated conditions. So in aerobic condition also nitrate reduction can take place, and this is particularly taking place in microvigula aerodenitrificans under air saturated conditions. So this bacterium use oxygen and nitrate as electron acceptors simultaneously. A similar property is also found in other bacteria, including Alkaligenes faecalis and Citrobacter diversus, Pseudomonas nautica, and Theospira pantotropha. So these are the microorganisms where you find that is the nitrate reduction in presence of air saturated conditions. Okay, so this is the uh, acrylate pathway which takes place in Clostridium propionicum. This is a fermentative nitrate reduction. So usually we start with the lactate. This is the chemical structure of lactate. So this is the L configuration form, and this is your D configuration form. So the lactate racemase, now lactase racemase converts the L form into the D form. Okay. Then the second enzyme, that is coenzyme A, transferase, it transfers the acetyl CoA. Okay. So what happens now? This is your lactyl CoA, then acrylyl CoA, and now this as coenzyme A, uh, See here, the coenzyme A is transferring to this acrylyl CoA to form the propionate. Okay. So the third enzyme, that is lactylyl CoA dehydratase. This is your third enzyme. That is the third enzyme. This third enzyme, lactyl CoA dehydratase. Are you able to see the? Okay. Now you are able to see the pointer. So this is lactyl CoA. In presence of lactyl CoA dehydratase, water molecule is given out and it forms the acrylyl CoA. Now this acrylyl CoA, in presence of acrylyl CoA reductase, this fourth enzyme, it forms this electron transfer takes place, and therefore ETF is unknown electron transfer factor. Probably it is NAD positive. So here it is NAD positive. This is accepting the electrons. Okay. And it is becoming a reduced. This is your oxidized one. Now, when it is accepting the electrons, it becomes a reduced one. Okay. So after that, what happens? This lactate dehydrogenase enzyme, fifth enzyme is the lactate dehydrogenase enzyme. So this in the oxidized state has become a reduced one. So particularly this lactate, it has produced a acetate, CH3CO, it is, yes, CH3CO, propionate. Okay. So this particular, okay, CH3CO CoA in presence of, just a minute, this is your pyruvate structure, CH3CO COH. Okay, so this is your pyruvate. So pyruvate, in presence of enzyme pyruvate ferredoxine oxidoreductase, you will get a acetyl CoA, and this acetyl CoA then gets reduced as the pi is removed out, CoA enzyme is eliminated in presence of phosphotransacetylase and acetate kinase in presence of the eighth enzyme, and okay, in presence of the eighth enzyme, that is transacetylase and acetate kinase, and you will get a acetate. See, the propionic acid producing bacteria, it ferments lactose, but while doing so, it is utilizing the nitrate, or it is reducing the nitrate, and instead of producing propionate, it is producing the acetate. लक्षात आली सायकल? मला काय म्हणायचंय की फ्रॉम लॅक्टेट हा बॅक्टेरिया जो आहे क्लॉस्ट्रिडियम प्रोपिओनिकम आणि मेगास्फिरा एल्सडेनी, हा बॅक्टेरिया लॅक्टेट युटिलाइज करतो आणि लॅक्टेट पासून जर का त्याला फर्मेंटेटिव्ह नायट्रेट रिडक्शन करायचं असेल तर तो कुठली सायकल ऑप्ट करतो? ही सायकल ऑप्ट करतो. तर युजली काय होतं, तो कंप्लीट सायकल इकडून इकडे इकडे, that is 2 3 4 4 then 5 6 7 आणि 8 ही सायकल ही अशी कंप्लीट करतो. अँड instead of making propionic acid तिथे बनत नाही. ओके, so this is the cycle. Then you have the another cycle which is called as the succinate propionate pathway, which takes place in propionate bacterium, which ferments lactate to propionate. तो बॅक्टेरिया लॅक्टेट फर्मेंट करत होता टू फॉर्म अ acetate molecule, and this succinate propionate pathway follow करतोय, and this propionibacter ferments lactate to propionate. Okay, so this lactate, this is the structure, that is CH3CHOH COOH is the structure of lactate. Now lactate in presence of lactate dehydrogenase gets converted to pyruvate. Okay, then from pyruvate you will get oxaloacetate in presence of enzyme methyl malonyl CoA, and that is pyruvate transcarboxylase. So this is your second cycle. So see here, the biotin carbon dioxide is used and biotin is given out and carbon dioxide is eliminated out. So this is the second one. Now from this oxaloacetate, from oxaloacetate you will have the formation of malate in presence of malate dehydrogenase, and then from malate you have the fumarate. Okay, in presence of enzyme that is malate dehydrogenase. Fumarase enzyme. From malate to fumarate is the this reaction is carried out in presence of fumarase enzyme. Okay. Then the fifth reaction is fumarate reductase. Means fumarate is reduced to succinate. So here you have the addition. See the double bond, carbon carbon double bond is now removed, and the CH you get CH2 here. So after that, the coenzyme A transferase is added. So coenzyme A is being added here at this location, and then in presence to form the succinyl CoA. Then the succinyl CoA is converted to the R-methyl malonyl CoA, and from R-methyl malonyl you will get S-methyl malonyl CoA. Okay, so this is the racemase structure. So there is just a configuration change. Uh, that is the stereochemistry is changed, and then in presence of the ninth enzyme, that is pyruvate dehydrogenase, here so the pyruvate is converted to acetyl CoA, and then acetyl CoA to that is CoA is removed, and the pi is added here, and then you get the acetate. So here you get the acetate, and then you have 11th one. Okay, 10th is the phosphotrans acetylase. This one, 10th is the phosphotransferase. Trans acetylase means the phosphate group is added here. This is the phosphate group being added here, and then the ADP is converted to ATP. So in this case, the function is that while doing so, the lactate is usually it is fermenting the lactate to propionate. It is giving you the propionate, one molecule of propionate. Also, with the help with synthesis of the propionate, you also get acetate, and also you get one molecule of ATP here. And since the NADH plus H plus is forming the NAD positive, so here you are getting three NADH plus H plus is oxidizing in the oxidized form, it is NAD positive. So this reduced one is getting converted to the oxidized. So it is using the electrons from the NADH plus protons. Okay, so this is how the that is the succinate propionate pathway takes place, which ferments the lactate to propionate and produce the ATP molecule. Okay, so hopefully you have understood the lecture. I will take you the to the another slide. So here I stop sharing, and then I will share the another screen. Mm, Wait a minute, I will. Okay. Yes, I'm taking your next lecture, that is on alcohol fermentation. My voice is clear now? Hello, कुणीतरी एक जण सांगा. Yes. हॅलो, आवाज येतोय?
Yes, ma'am.
ओके, चलो. स्लाइड दिसतीये का? एकदा कन्फर्म कर.
नाही, ma'am.
नाही दिसत आहे का? एक मिनिट.
Yes,
आता
ma'am. दिसतीये.
दिस
हो, ma'am.
दिस ओके. आता सांगा.
Yes, ma'am. दिसायली.
दिसत आहे ना? ओके. So I will start now. So the next point is alcohol fermentation. Usually the alcohol fermentation is called as a ethanol fermentation, but in alcohol you have many types like ethanol, butanol, propanol, etc. But when we speak about the biofuel or a fuel, we only speak about the ethanol. So ethanol fermentation is also called as a alcoholic fermentation. It is a biological process which converts sugars such as glucose, then fructose, sucrose, and when it is converting or when it is metabolizing these sugar molecules, usually in the cell, that is, you know that glycolysis takes place in the that is, if you consider a prokaryotic cell, you will see that the glycolytic pathway takes place in the that is cytoplasm. So this metabolic pathways which takes place in the bacterial cell or any eukaryotic cell is helpful for making energy in the form of ATP. So in this glucose sugar or the sugar molecules, they are converted into cellular energy, and this then they are producing the ethanol and carbon dioxide as the byproduct. So carbon dioxide is the byproduct. So we have many yeast like Saccharomyces cerevisiae which performs best when there is absence of oxygen. So usually the fermentation process takes place in the anaerobic condition, and therefore we call it as a anaerobic process. So it also takes place in some species of fishes also like goldfish and carp, where the lactic acid fermentation takes place. In our muscles also, what happens? The lactic acid, that is lactose, is converted to the lactic acid, so which provides the energy when there is a scarcity of oxygen, or when the oxygen amount is low in the muscles. So ethanol fermentation has many uses, including the production of alcoholic beverages and the production of ethanol fuel also, and also for the bread making. So if you observe the biochemical process of fermentation of sucrose, now we are going to see the alcohol fermentation by taking two sugars. First is the sugar, that is sucrose molecule, and second one by taking the glucose molecule. So glucose is very famous. That is, glucose is sugar which is usually consumed by the prokaryotic cell and is available in their environment when you are adding glucose in the media also. So but this sucrose is present in the cane sugar, and the chemical equations which undergoes fermentation of sucrose C12H22 O11 into ethanol, that is C2H5OH. So here you will get mole of glucose and 2 moles of ethanol and 2 moles of carbon dioxide, producing 2 moles of ATP molecules. So you will get 2 ATP molecules in this process of fermentation of sucrose. So then that is C12H22O6 from C12H22 22O11, sorry, O11, you will get 1 mole of glucose. So this 1 mole of glucose is then again further can be converted into alcohol, that is ethanol, with the 2 carbon dioxide molecule. So sucrose is a sugar composed of glucose, which is linked to the fructose. So in sucrose, you have one aldehyde containing sugar moiety, that is sugar molecule, that is glucose, and the another one which is having a ketose group. So in the first step of alcoholic fermentation, the enzyme invertase cleaves the glycosidic linkage. So here it is cleaving the glycosidic linkage in presence of invertase enzyme between the glucose and the fructose. And now you will get 1 molecule of glucose and 1 molecule of fructose. Okay. Then the fructose is converted to glucose. Now you have got 2 molecules of glucose. Now next, each glucose molecule is broken down into 2 pyruvate molecules, as we have seen in the glycolysis pathway. So this glycolysis is summarized by the equation, that is C6H12O6 plus 2 ADP plus 2 Pi plus 2 NAD positive. That this is your NAD reducing agent. So here you have got the reduced NADH plus H plus and 2 water molecules with formation of pyruvate and 2 ATP molecules. Now these pyruvate and inorganic phosphate you will get here and in Pi is the inorganic phosphate. So finally pyruvate is again converted to ethanol. So in Krebs cycle, you will see that pyruvate is what we say, pyruvate. From pyruvate, you get the acetyl CoA, and this acetyl CoA combines with the citric acid to form the oxaloacetate, and it is moving in the Krebs cycle. But in the fermentation process, in the alcohol fermentation, the pyruvate is not entering into the Krebs cycle. Here the pyruvate is getting converted to ethanol and carbon dioxide in 2 steps, regenerating oxidized NAD positive, which is needed for the glycolysis. So 1 molecule of pyruvate in presence of pyruvate decarboxylase, it gives you CH3CHO. Okay, you get this particular compound, and then this CH3 CHO in presence of reducing agent is converted to C2H5OH, that is the ethanol, plus NAD positive in the oxidized state. So this reaction is catalyzed by the alcohol dehydrogenase enzyme, which you find in the Baker's yeast or in the Saccharomyces cerevisiae. So this is your alcohol dehydrogenase 1 enzyme, which is usually found in Saccharomyces cerevisiae. So only 2 steps are required for the alcohol fermentation. One is by means of that is pyruvate decarboxylase enzyme, which catalyzes the conversion of pyruvate into, that is acetaldehyde, CH3CHO. Okay, so you have this, and then later on the acetaldehyde in presence of NADH plus H plus is converted to ethanol. So as shown by the reaction equation, glycolysis causes the reduction of two molecules of NAD positive to NADH, and therefore two ATP molecules are also converted to two ATP molecules, and two water molecules are given out via substrate level phosphorylation. So here you get two molecules of ATP. Okay. So Escherichia coli is also capable of fermenting arabinose. Arabinose is also a sugar molecule to and this arabinose is fermented to ethanol, but it is not naturally tolerant to high ethanol levels. So just it makes a few that is few amount of ethanol levels, and then it limits its production. Another bacteria, because it cannot tolerate the high concentration of ethanol. Another bacterium, Zymomonas mobilis, is naturally tolerant of high levels of ethanol, but it cannot ferment the anaerobic, that is arabinose. It is highly tolerant to uh, tolerant to ethanol, but it cannot ferment the arabinose sugar. Zymomonas mobilis uses slightly different pathway for ethanol fermentation. So Zymomonas mobilis is utilizing different pathway. As a result, the expected ATP yield is only 1 ATP per molecule of arabinose. From glucose you are getting 2 molecules of ATP. But when arabinose sugar is used, you will get only 1 molecule of ATP. Yeast and other microorganisms, they ferment glucose to ethanol and carbon dioxide rather than lactate. So the yeast and microorganisms, they are using the glucose molecule and not the lactose molecule. So glucose is converted to pyruvate by glycolysis, and pyruvate is then converted to ethanol, and carbon dioxide is given out in two steps, which we have seen. Pyruvate is first decarboxylated in irreversible reaction, which is carried out by enzyme pyruvate decarboxylase, and then this reaction is simple decarboxylation, removal of the carbon dioxide, and it does not involve the net oxidation of pyruvate. And then this pyruvate decarboxylase enzyme, it requires Mg2 plus ions, and also it is very tightly bound to the coenzyme, that is TTP, that is thiamine pyrophosphate. So usually this is depicted in the figure, that is glucose is first converted to two molecules of pyruvate, and then pyruvate is converted to acetaldehyde with eliminating two molecules of carbon dioxide. So this is your glucose converted to pyruvic acid, and then the pyruvate is converted to acetaldehyde in presence of pyruvate decarboxylase, because there is removal of two molecules of carbon dioxide. Then the two acetaldehyde in presence of the third enzyme, that is we call it as acetaldehyde or alcohol dehydrogenase 1 enzyme, is converted into two molecules of ethanol. So the oxidized form of NAD is getting converted into NADH positive, and this NADH positive is then again in seconds in first step, the NADH positive in the oxidized state is getting reduced to NADH plus H plus, and two ATP molecules are synthesized by the first step because of substrate level phosphorylation. And here what happens when the 2-acetaldehyde is uh, getting converted to ethanol, the reduced NADH is again uh, mm, oxidizing the NAD positive, and this NAD positive, which is in the oxidized form, can be reused for formation of the pyruvate. So this circle is going on. Okay. So in the second step, acetaldehyde is reduced to ethanol through the action of alcohol dehydrogenase, and with the reducing power furnished by NADH positive derived from dehydrogenation of glyceraldehyde 3-phosphate. So from here you are getting the that is reduced NADH positive, and this NADH positive is utilized by this alcohol dehydrogenase enzyme, so that it will convert acetaldehyde into ethanol. This reaction is well studied in the case of hydride transfer from NADH. I will show you in the next figure. So ethanol and carbon dioxide are thus the end product of ethanol fermentation, and the overall reaction is given as this. So in presence of magnesium ions and thymine pyrophosphate, pyruvate is first decarboxylation takes place, and from pyruvate you will eliminate out the carbon dioxide to form the acetaldehyde. And in presence of alcohol dehydrogenase, the acetaldehyde gets converted to ethanol. And this is the step, that is the NADH reducing power is supplied from 3-glyceraldehyde phosphate, and oxidized NAD positive is made. Okay. So this is the anaerobic conversion of glucose to ethanol via glycolysis in alcohol fermentation. You can make alcohol from malt, you can make it from jawar flour, you can make it from rice, you can make it from grapes, anything, but you will get the alcohol fermentation if you employ the Saccharomyces cerevisiae yeast. And this is your distillation unit. When it is distilled, the pure alcohol or the distilled alcohol comes out. So this is alcohol dehydrogenase enzyme. How it is linked, as I have said, it requires Mg2 plus ion, and it requires the hydride ions. So there is a transfer of the hydride ions from the NADH. So zinc is at the active site, which for zinc which is present at the active site. This molecule, this is your enzyme. This structure is your enzyme, and this is your active site. So this is your active site. So on the enzyme you have the active site, which play an important role in catalyzing that particular reaction. So the zinc is at the active site, which polarizes the carbonyl oxygen of the acetaldehyde. So substrate is being added to this here, allowing the transfer of hydride ion from the NADH. So from NADH, this particular hydrogen is accepted, and it comes to this location. The reduced intermediate acquires a proton from the medium, that is where it is located, that is the blue. Okay, and to form the ethanol, so you will get the ethanol. See here, that is H has come from the proton, and this H has come from the hydride ion, this hydride ion of the reducing agent. Okay, so the alcohol dehydrogenase is present in many organisms that metabolizes ethanol, including the humans. In the liver, it catalyzes in humans. In liver, it catalyzes the oxidation of ethanol, either ingested or produced by intestinal microorganism, because intestinal microorganisms also produces the alcohol. Also, whatever the human beings are intaking the alcoholic beverages, that also. So with concomitant reduction of NAD positive to NADH, uh, it is catalyzing the oxidation of ethanol. So in this case, the reaction proceeds in the direction opposite to that involved in the production of ethanol. So when you are consuming alcohol, not you, when somebody is consuming alcohol, or when the small amount of alcohol is produced by the intestinal microorganism, exactly the opposite steps is taking place. That is, ethanol in presence of NAD positive then gets converted to NADH plus NADH, and you will get the pyruvate. This is the opposite reaction which is observed in the liver, and it is removing the alcohol. So okay. So, as in lactic acid fermentation, when the lactic acid fermentation takes place, when lactose is used, that is, the lactic acid is used, not lactose, lactic acid is used, there is no net change in the ratio of hydrogen to carbon atoms when glucose is fermented to 2 ethanol and 2 carbon dioxide is given out. So here HC ratio is 2, as well as here H and C ratio is 2. So in all fermentations, HG ratio of the reactants and the product remains the same. So pyruvate decarboxylase is present in the brewers or the baker's yeast, that is Saccharomyces cerevisiae, as well as in other microorganisms that ferments glucose to ethanol, including some plants. So carbon dioxide, which is produced by the pyruvate decarboxylation process, is responsible for characteristic carbonation of the champagne. Uh, you know the champagne? It is a beverage which you usually observed in on the playground of that is cricket. जेव्हा क्रिकेटची मॅच कोणी जिंकतात तेव्हा शॅम्पेनची बॉटल काढतात आणि त्याचं झाकण कॉर्क ओपन झाल्याबरोबर तो फेस आणि फवारा बाहेर निघतोय. It is only because of this carbon dioxide. The ancient art of brewing beer involves several enzymatic processes in addition to the reactions of ethanol fermentation. So, in baking, also, carbon dioxide is playing an important role because your bread uh, is made to be fluffy, spongy होतोय आणि ते कशामुळे होते? It is because of the accumulation of carbon dioxide. Carbon dioxide. So it ferments the sugar, that is glucose and lactose, and then the dough म्हणजे तुमचा आटा तो राईस होतो because of the elimination of carbon dioxide. So this enzyme is absent in the vertebrate tissue and other organisms that carry out the lactic acid fermentation. Now we will see the another concept that is related with the alcohol fermentation, that is called as the Pasture Effect. So when any glucose molecule or any sugar is converted to ethanol, usually it is linked with the Pasture Effect. Now this effect was discovered in 1857 by Louis Pasteur, who showed that aerating yeasted broth causes the yeast cell to grow and to increase, while fermentation rate is decreased. Means what happens when the yeast broth is aerated, yeast broth is aerated, yeast cell ची ग्रोथ होते, म्हणजे ग्रोथ इन्क्रीज होते, पण अल्कोहोल प्रोडक्शन हे कमी होते. And that was proposed by Louis Pasteur. And then later on, Louis Pasteur was selected to the Academy des Sciences in 1862 because of this invention. And then he became the professor of geology, physics, and chemistry at École des Ducs Arc. Okay. So what is this effect? This effect is explained that as when the yeast is growing and it is undergoing a alcohol fermentation, usually we require that the yeast should continue with the formation of ethanol. But sometimes what happens when the oxygen concentration begins to increase, usually the uh, alcohol production is decreased and the growth of the yeast is increased. So that effect is called as the Pasture effect. So as you know that yeast is a facultative anaerobe, they produce energy using two different metabolic pathways. One pathway is when there is a oxygen concentration is low, the product of glycolysis pyruvate is turned into ethanol. So when concentration of oxygen is very low, it is converting the pyruvate to ethanol and carbon dioxide. But when you have the oxygen amount in higher concentration, oxygen in higher concentration, then it undergoes the conversion of pyruvate to acetyl CoA. So here in ethanol production, you will get 2 moles of ATP molecule per glucose molecule. But if there is oxygen concentration more in number, then the pyruvate is converted to acetyl CoA. And that is usually done in the Krebs cycle or citric acid cycle. And here it increases the efficiency of ATP formation, and here you will get near about 31 to 29.5 moles of ATP per molecule of glucose when it carries out the glycolysis as well as the Krebs cycle in detail. So when the oxygen is present, you will get this amount of ATP, and when the oxygen concentration is low, you will get ethanol and carbon dioxide with only 2 molecules of ATP. So the yeast decides which type of metabolic pathway has to be taken place, whether depending on the oxygen concentration, whether to go for ethanol production or whether to go for that is the Krebs cycle, glycolysis and Krebs cycle. So it depends on which shuttle is used for reducing the reducing equivalent. So in this case, you will get the reducing equivalents in the form of NADH, which is formed in the cytosol. So therefore, about 15 times as much glucose must be consumed anaerobically as aerobically to yield the same amount of ATP. So in order to go for glucose metabolism, 15 times is required glucose to be consumed anaerobically, because in aerobic pathway you get 31 ATP molecules. Okay, but in this case, if it is undergoing an anaerobic pathway, then it is then it is synthesizing only 2 ATP molecules. So if you want the same amount of ATP, that is say for example 30 ATP, 31 ATPs or 29.5 ATPs, means you have to go for 15 times, means 15 into 2 you will get 30 ATPs, means for same amount of ATP 15 times it has to go anaerobically, and this is not feasible. So under anaerobic conditions the rate of glucose metabolism is faster, but the amount of ATP produced is very smaller, that is only you will get only 2 ATP molecules in the anaerobic process. Okay, so when exposed to aerobic conditions, the ATP and the citrate production increases and the rate of glycolysis slows, but ATP and citrate produce act as a allosteric inhibitors for phosphofructokinase 1, which is the third enzyme in the glycolysis pathway. That is conversion of phosphofructokinase to glyceraldehyde 3 phosphate. Glyceraldehyde. That is glucose is converted to glucose 6 phosphate. Glucose 6 phosphate is converted to fructose 6 phosphate. Fructose 1 6 phosphate is converted to fructose 6 phosphate is converted to fructose 1 6 phosphate. And this is the enzyme that is which is catalyzing the conversion of fructose 6 phosphate to fructose 1 6 phosphate diphosphate. Okay, so this third enzyme in the glycolysis pathway is inhibited when the when there is a aerobic condition, and the rate of glycolysis is slowed down because of the inhibition of this particular pathway. So the Pasture effect will only occur if glucose concentrations are low. So when you will observe this Pasture effect, yes, and any problem? Do you have any problem? So Pasture effect will only take place when glucose concentration are low. Just Vaishnavi. Yes, do you have any problem?
No, ma'am.
Okay. Where is my slide now? Okay. So when the concentration of glucose is low, that is less than 2 gram per liter of the media, uh, and if nutrients, mostly nitrogens, are limited, then you can see the rate of glycolysis is slowed down. So from the standpoint of ATP production, it is advantageous for yeast to utilize citric acid cycle in presence of oxygen, as more ATP is generated from less oxygen. So Bolton et al. In 1996, they maintained that have mentioned that yeast will follow the anaerobic. This is not maintained. Have mentioned MEN. TIONED mentioned that yeast will follow the anaerobic pathway rather than aerobic fermentative pathway if glucose is not limited. In that respiration, it is capable of producing more ATPs than glycolysis per molecule of glucose. So it is ATP, main aim of an organism is to produce the ATP, because ATP is the energy source. So when to go for fermentative process, or when to go for glycolysis, or when to go for glycolysis plus Krebs cycle, it decides on the concentration of the glucose molecule and the oxygen concentration. Also, it requires more energy in terms of enzymatic and mitochondrial requirements. So thank you. Uh, Here I am stopping. Just I will take your